Do you feel glamorous in your Tyvek suit?
No.
That's what I'm wondering.
No.
Only when he puts on a tiara on top.
That's right. I actually draw one on my Piper hood that I wear.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klonowski.
And I am David Peterson, your host for today's episode. Today's guest is our first to have had a full FBI background check. Explanation to follow.
Our guest today is Dr. Jeff Hogan, professor in the Department of Infectious Diseases and the Department of Biomedical Sciences. Jeff, welcome to the podcast.
Thanks for having me.
So what we really like to start out with is to find out what brought you to science. And we've been asking this of our guests lately, and they all have really interesting and different paths to their careers. So how did you end up here at UGA?
So, how did I end up here at UGA?
You can start back in high school or wherever you need to.
Well, I got my first microscope when I was 6 years old for Christmas.
Ooh, I don't think we've gone back this far yet.
And the die was cast.
It was, and it was all over with crying at that point, I think. So every bug, anything I could find, I put under the microscope and kind of never looked back from there. So after high school, I went to college. And I originally planned to go to medical school, but I decided I really didn't want to do that. I became interested in research after a microbiology class that I took, a course in college, and actually went to a seminar about immunology and decided, that might be really interesting. Went to Department of Microbiology at University of Mississippi Medical Center. Unannounced, made no plans, walked in and said, hey, can I talk with the department chairman? I didn't even know his name. I mean, there's no internet back then, right? So, how do you look that up? And I dressed kind of like I am today in jeans and stuff. And he was kind enough to sit down and talk to me for 2 hours. And by the end of it, he said, you know what? I'll give you a shot. And I made it through, and then took a postdoc at St. Jude in Memphis with David Woodland. And that was great. Went up to Trudeau Institute. And then from there, went to USAMRIID. I had developed an interest in biocontainment, especially viruses, Ebola virus in particular. Got an offer to go to USAMRIID and run a lab there. I did. And then, of course, biodefense became a big thing after the anthrax letters, establishment of federal select agent program. So got recruited down to Southern Research Institute in Birmingham. And by that point, I'd gone from academia to government, USAMRIID, to industry in Birmingham, and decided I really wanted to go back to academia. And UGA was opening up the Animal Health Research Center to do biocontainment research. And as soon as I saw that, I said, that's it. I really want to go there because of the capabilities that it gave me. So that's how I landed here.
You know what I find most interesting and compelling about that story is this department head who unannounced, no appointment, sat down for 2 hours with someone that just walked in. He took the time. I would say that 2 hours has paid off really well.
It's paid off really well.
Yeah, it's helped me raise 2 kids. But no, it's been a great ride. And, I didn't even realize at the time that department was world-renowned for comparative immunology.
Right, because you started in fish, correct?
Exactly. Just like another faculty member here, actually a couple of them, and several of our competitors were actually in the Department of Infectious Diseases. So I got to interact with them. They were the first people that I met at the first scientific meeting that I went to, were faculty members that when I came here, I joined the faculty and went to faculty meetings with them. So it kind of came full circle.
So from competitors to colleagues.
Yeah, absolutely.
Yeah, it can happen.
That's right.
So Jeff, why don't we jump to the present day? What's the focus of your current research?
So I did a lot of work on vaccines, and it was really interesting. But over time, as I taught a cell biology course to the vet students, I learned a lot more about cell biology and became more interested in host pathogen interactions. And kind of immunology went to the side a little bit because I'm teaching cell biology, I'm getting into it. And I started looking at therapeutics. I'd done a little bit of therapeutic development for the first coronavirus outbreak in 2003 when I was at Southern Research. And I really wanted to get back into that. So now we're really focused more on therapeutics for viral and bacterial pathogens. And that's focused mainly on those that require BSL-3 or BSL-4 containment.
Well, I think this is a good time to ask about the difference between the different levels of laboratory biosafety classifications. So I'll start off with my interpretation of BSL-1, which is basically my kitchen.
I think you're absolutely right. Basically a bench top, a counter, and then you put a sign up that says this is a lab.
So would BSL-2 be you just got to maybe wipe things down a little bit more? Maybe a little bleach, maybe a little UV light?
Yeah, disinfection, maybe a biosafety cabinet, especially if you're working with like flu, influenza, for example. I think that's a really good one, like a standard, run-of-the-mill circulating strain of flu every year would be a BL2 level.
Right, not the bird kind.
No, not the avian kind. So,
Okay. So now we're moving up to 3.
yep, BL3. In my opinion, you would think BL4 would be the most dangerous. My opinion, BL3 is actually the most dangerous containment level. So BL3, an example would be like SARS-CoV-2, CoV-1, strains of influenza, like avian influenza strain, the currently circulating H5N1 that's been isolated in cattle. That's BSL-3 pathogen. Yeah, so those are some kind of more well-known examples, I think.
But this is where we get into the FBI background check.
Yes.
In order to work at BSL-3, they want to know you're a good guy.
Oh, yes. So I've been fingerprinted many times, both outside of work and at work. We won't talk about that. But no, and being serious about it, they do extensive background checks, everything from, national database checks. They do, of course, take your fingerprints and check those. Psychological evaluations. There's a lot of credit checks. I mean, it's very, very extensive. And the reason for that is, you're working with pathogens that actually infect, for the most part, infect humans and animals too, which could cause a huge outbreak of disease in animals like we're seeing again with H5N1. But they want to make sure that the people that are working with the pathogens are vetted, they're responsible, and they're stable, right? That, as I mentioned before, the anthrax letters that were mailed out in 2001, that was when I was at USAMRIID. And that actually led to the creation of a lot of the regulations that are in place now, like the background checks and things like that. Before then, none of that was in place. So that was put in place by lawmakers. In some ways it's good, some ways it's not. It slows down research. But again, I think it instills a little bit more confidence from the public in what we do, that we know how to handle. We've been vetted.
And that's continual. It's not like a one-time up front. You're kind of, tested and monitored.
Every year we go through the same background checks to make sure nothing has happened that we haven't reported. And people have been, for example, they've gotten a traffic ticket or they've gotten some kind of citation or arrest, and that does come up, and then they are, that's dealt with, whether they're put on probation in the program or whether they're removed from the program. So that's a yearly thing. And then training, of course, is annual. And it grows every year. It gets more and more extensive. And that kind of gets to the good and the bad. It's good that we have these checks and balances, but you can imagine you have training for respirators, you have training for medical evacuations, you have all these different training requirements, and that eats up a lot of the time. It also makes it difficult to get students. We can't have undergraduate students in those positions, because by the time we train them, it takes 6 to 9 months. By the time they go through all this, their rotation be over. That semester would be over.
So you kind of mentioned a few things here and there. So things like the respirator. So what types of precautions would one normally be taking in a BSL-3 facility?
So we use PAPRs. That's basically a HEPA-filtered respirator that's battery powered. and you check those every time you go in, and those, they work very well. As I mentioned before, kind of in my opinion, BL3 is slightly more dangerous than the BSL4 environment. And that's because in a BSL4 environment, you're completely encapsulated. So you're in, think of it as a hefty bag suit with an air line from an air compressor literally attached to it.
And it looks like a spacesuit.
Yeah, like a spacesuit. So you're completely encapsulated. Whereas in BSL3, your neck may be uncovered. You can wear full, headgear that will cover everything. But again, you're not sealed in. So, and you're not wearing a thick, heavy, durable suit. You're wearing Tyvek type material, and it can rip and tear and things like that. In BL4, you'll have 3 or sometimes 4 layers of gloves on, where we wear 2. So, that's something that, I make sure, especially with graduate students or with postdocs, if they don't have containment experience, that they know how to use the equipment. They're very familiar with it. They know what to do in the event, let's say a battery, you lose power or something like that. But again, that's where all the training really comes into play. You have to know where things are. You have to know what to do in the event of an issue, and not, what's the right way to put this, to maintain your composure, right? Not become afraid of the situation. You have to be able to think clearly under kind of pressure. I mean, that's really pressure for yourself and the other people in the lab.
I would guess that you'd also have to be sure that you don't become kind of blasé about it.
Absolutely. So that is, I believe, the biggest reason for the annual training is it brings that back to the forefront. You don't think about a lot of things that are brought up in the training. You kind of go, yep, that's right. And then you kind of push them aside. And over time, you become complacent, of course. And that kind of reels everybody back in a little bit. And we try and stagger those trainings through the year, right? So we may do some let's say February, and then we've got some in March, and then we've got some in May, and we kind of spread these along. So it's constantly being reinforced in your mind that you need to be safe.
So we've talked some about the BSL-3 conditions. What makes an organism, what's the characteristics of an organism that relegates it to BSL-3 research?
That's a great question. So really it comes down to, are there any therapeutics or vaccines available for those pathogens? And then also the ability to transmit from person to person. So usually anything in a BSL-3 or a BSL-4 environment, you're going to have very few options when it comes to treatment or for vaccination. And it's slightly increased when it comes to BSL-4 pathogens, so setting 3 and 4 apart. You may have some antibiotics that have some efficacy, for example, against a BSL-3 pathogen, but, then again, let's take Bacillus anthracis spores, probably better off to be worked with at BSL-4, although you don't have to, but to contain those and not allow them to spread outside, right? That's a huge component of what we do. Right now, the lab is completely shut down for recertification of all of the filters in the building, all of the biosafety cabinets, all the equipment that we use. Every piece of equipment, every monitor, camera, everything is thoroughly gone through at a great deal of cost every year. And those documents, when we are inspected by the CDC and USDA, they review all those documents and make sure that everything is up to par and that we're good to go. And we have, knock on wood, we've not had any major malfunctions.
I've seen the room at the ARC that has all of the stainless steel piping that carries, fresh air, filtered air, exhaust air, which is also filtered, we should say, before it's released, and then all the water. It's a huge space. It's larger than the BSL-3 labs that are on that first floor, and you go to the other floor, and there's a lot more space, but yeah, it's like half the building is on the stuff to make sure that the other half of the building is safe.
It is. I like to tell people that think of the lab space as a third of the building, where 2/3, both a third of it above and a third below in the basement, are used to basically operate that one, area of individual labs. So yeah, it's a huge undertaking, a lot of moving parts. I don't know the exact amount of money. I believe it's a little over $100 million went into that building overall.
Overall.
And multiple iterations, low cost bidder. We won't get into that. But I've had plenty of arguments with architects. That's not going to work. Value added engineering or something of that effect. But yeah, the facility now is, as of about 10 years ago, we have the capability to work with everything from cattle to horses. We've done H5N1 infections in cattle in that building, which has been, I mean, really, not to kind of jump around, but if you look at the planning for the Animal Health Research Center, that was 50 or 60 years ago. And most people don't realize that there was a lot of forethought that was put into building that. And why would you do that as a vet school is usually the second question. It's, well, zoonotic pathogens, those that jump from animals into humans. So the dean and others, Keith Prossie and others, had the foresight to lobby the state to try and get the funds to build this just in case of an outbreak like SARS-CoV-2, which that, of course, is humans, or H5N1.
Ebola, all of those things.
Now, of course, they will not let me convert any of the rooms to BL4. I've tried. They don't want that liability, and I can certainly understand it. But yeah, I mean, I think that, the foresight was great. And really, when you look at the most recent pandemic in 2020, the amount of research that was done there, I mean, we're talking therapeutics that went into individuals, that went into humans, right? Vaccines that went into humans, however you feel about the vaccines, regardless. The capability to do that level of research in an academic environment is incredibly rare. There are very few of them in the country, let alone around the world. So hats off to them. Like I said, when I was looking to go back into academia from industry, kind of made that trifecta, that facility was critical. I said, I'm not going to find that anywhere else. And yeah, up until I believe Kansas State built one. Up until then, there wasn't anything like it in the country.
Being able to swing into the action when a pandemic starts sounds really glamorous. I think someone should make a movie out of that. They probably already did. But is the day-to-day work? Do you feel glamorous in your Tyvek suit?
No.
That's what I'm wondering.
No.
Only when he puts on a tiara on top.
That's right. I actually draw one on my Papper hood that I wear. But yeah, I always tell people they watch a movie Outbreak or something like that. And I go, I don't know how to fly a helicopter, can't synthesize 100 kilograms of antibodies in a day. It's hot, it's cumbersome, it's tiring. I think I've probably lost half my hearing from a respirator blowing past my ears over the years. Been doing this work for 25 years now. I was thinking about that on the way over. I was like, I can't believe I've been doing this work for 25 years. But, it's dirty. So I tell people it's definitely not glamorous. You really have to have a passion for working with these pathogens, and you have to respect the pathogen. But you can't be afraid of it either. Or I've seen people get paralyzed in containment labs out of fear. Right? They have a hole in the glove, these things happen. And if you don't have the right mindset, it's really not for you. It's not for everyone.
Yeah, it makes me think about the personality of people who work in biocontainment. Like, are biocontainment scientists like people who scuba dive also and skydive and who are just kind of good at having those types of hobbies?
I think some are. I would contrast myself to Eric LaFontaine. Collaborated with Eric for over 15 years now. He's very mild, he's very unassuming, he's very quiet, whereas I'm kind of just the opposite of that, which I think actually is really good when it comes to students and postdocs and training them in that environment. They need kind of both sides of that, right? They need to be able to see that you have to get in there and you have to catch this animal that's infected with something that could potentially harm you, right? But at the same time, you have to have that level of composure. You have to be able to go out, grab that, and handle it, take your samples, etc. But you have to maintain that composure level too, because remember where you are, remember what you're working with, remember what the dangers are. So it's a good balance. I have most of the work that is actually done in the containment labs in the ARC are collaborative research projects, and I think for that reason.
Right. So I think I'm the right person to ask this question. So you went from doing basic immunology to studying some serious infectious diseases. Is that because you kind of anticipated this is where like all of the research was going to go in terms of human diseases? Is this because you did think it was glamorous and now you know it's not glamorous? Or what kind of drove that jump? I'm just curious.
So I always had an interest in containment work, and that had nothing to do with a movie. I was thinking about this again this morning. There was no internet. When you had to go pull a manuscript or papers or whatever, you went to the library, you used a photocopy. You had a cart with journals you'd stack up and use a photocopier. I mean, it's kind of like ancient times.
Yeah, you had to worry about the crease in the center and making sure you got it flat enough.
My abacus, I had to get that.
I just lost all the Gen Z.
Exactly. They're gonna be Google that. But no, I came across I was doing a journal club, came across a couple of articles about Ebola virus, and the first thing out was, it's 90% fatal. And immediately I was intrigued. I was like, okay, so what is it about the immune system that can't handle this infection? Because I had never really thought about it that way. And I became intrigued with it. So I reached out to CDC when I was getting ready to look at postdocs, and talked with a couple of individuals there. They've since retired now, and tried to get an ASM fellowship and wasn't able to do it. But there I was going to develop animal models for Ebola virus, because I had extensive experience as a comparative immunologist with everything from fish to birds and everything kind of in between, mice, even some human experiments that we were doing to compare, basic cellular responses. So I had that interest, but it kind of went away. Did my postdoc, had a great postdoc working with influenza. Little did I know I was going to be putting the BSL-3 at St. Jude. And so I kind of got a taste for it there. And I said, you know what? I like it. No one else really wants to do this. I'm in here by myself. I have the whole space to myself. Came across one of the first H5N1 isolates, HK98, was about a year after I got there, and did my postdoc. Actually had someone reach out to me from USAMRIID. They'd seen a presentation that I'd given at a national meeting looking at T-cell responses, and they were interested in studying T-cell responses to Ebola virus because they had a couple of vaccine candidates coming online. And I went down, interviewed, saw the place, and I said, absolutely, I want to come here. And I was told, and this was granted 2000, late 2000, before biodefense was a big thing, you are going to ruin your career. That was what I was told. If you go there, it's going to ruin your career. It's never going to go anywhere. You're going to be in a government system, et cetera. And little did I know, about 6 months after I got there, the anthrax letters were mailed out, which just kind of ripped the whole biodefense field wide open. And it was just fortuitous. It was absolutely fortuitous. I had no idea that the office, like 100 feet down the hall from where my office was, they think was where some of those letters were put together and mailed out. So
what you're telling me is that while dangerous pathogens cannot escape a BSL-4, a dedicated researcher can.
Yes. Lots of things can happen. And that gets comes full circle to the personnel component and why we have those checks and balances in place. And that, again, is a good thing. It's pretty simple. Most of the threats that we have are not really stolen vials or anything like that. It's an insider, disgruntled, personnel kind of approach, and that's been found to be the case with a lot of them. So again, it's and that's why you don't have many people. So we generally have a small group of very dedicated individuals that we are very close with. So we will know if something, and they're very good about telling us if something's going on. So support's key.
So I have a question. 2020, SARS-CoV-2 comes along. You'd been around for the first SARS outbreak. Looking back, did you think it would be this virus, that a SARS-CoV virus would be one that would kind of stop the world in the most recent global pandemic?
I really thought it would be flu, but I did keep my original isolates from 2003. They put SARS, but then it was just SARS coronavirus.
Right.
It wasn't COVID-1, but we'll call it COVID-1. They put it on the select agent list, which requires additional, training and documentation and background checks and things like that. And I went through and it's a lot of paperwork. It is a lot of stuff that you have to deal with. I kept them, as I remember someone saying, and I watched funding die off as a result of this. Someone said, "Well, we'll never have an outbreak of this ever again." And of course, NIH quit funding it. But I kept the isolates, and I was like, you know what, it's going to come around again. But no, I actually thought it was going to be influenza. I became a very popular individual around the beginning of 2020 when people kind of realized this was going on. I actually knew that this was happening. I believe it was November of 2019. I got a phone call from someone that was doing surveillance in China. I'd maintained contacts with a bunch of people over there, and they said, yeah, it's happening again. I said, it's not right. That can't be happening again. And they swore Wuhan was already, on the way of being locked down, or that was in process. And so I started looking at retirement. I was like, that's it. I've had enough. 20 years at that point of this. I'm just gonna retire, and but I couldn't keep my health insurance. So I got to be here 25 to keep your health insurance. So.
And if there's a pandemic, you kind of want that.
Yeah,
Exactly. You don't want to lose your insurance or get that cut off, especially with a couple of kids and going into a pandemic. So I was just going to sit at home like everybody else. I was looking forward to it.
So Jeff, over your career, you've worked in a lot of different sectors. So you did academia, you did government, you did industry. So for any grad students who are listening, what are some of the kind of pros and cons of those different entities?
That's a great question. And I have talked with so many students about this over the years. They are very different. I mean, again, the science is the science, right? So the difference really is in academia, you excel based on your own experience and on your abilities, so you have freedom there. When you go into government, usually the science is driven by a mission, right? So what is the mission? So for example, at USAMRIID, my mission was to measure T-cell responses to filovirus infections, Ebola and Marburg, primarily in the context of vaccinated animals using these animal models. And, it never mind the fact that the vaccine candidate that they had didn't work. So my mission was impossible, which kind of led to me going into industry. But it's really mission driven. So if you don't like the mission, there's not really anything you can do about it. So you lose some of that freedom, right? But you generally have a steady level of support. So for example, getting funding for, particular projects, as long as they fall under the mission, is much easier than in an academic environment, right? Where you have to basically fight for NIH or DOD or USDA or whatever funding agency, is kind of in line with what you're working on. So, there's that benefit of it too. It's usually more stable as well. And then when you get into industry, really, you don't control the mission, and you don't really control the experiments. You're basically you're a hired gun. you get a project that a group, for example, a small startup biotech company needs your expertise to, screen against their compounds against some virus, a panel of viruses. Well, you don't really have control over that. Now, the upside, though, is generally in the industry, they put golden handcuffs on you. So the pay's really good. And, you get to meet a lot of different people too. I actually, probably the best thing about it was I learned the business of science, the actual money, dollars and cents. So if I kind of look at all three of those, I learned how to be a good scientist. I had some amazing people train me that I was able to work with, outstanding individuals. Laundry list of, 5 pages. So I really learned that in academia. Then when I went to USAMRIID, I learned the government system, which of course most of our funding comes from the government system. And I learned biocontainment at the maximum level that we have. So that was, I like to think of it as kind of, adult level biocontainment, if you will. And then when I went into industry, of course, I'd go to lunch with the accountants and they would explain to me cost of money and how to budget these and really be competitive, what indirect costs are, what are they used for, intellectual property rights, and basically, again, the business of science. So it really gave me a well-rounded view of it. And so basically, myself with Eric, we run the labs as businesses. The difference is the products are students or postdocs become faculty members, data sets, papers, presentations, things like that. So it's still run as a business. I don't think a lot of people realize that UGA, the vet school, all the above, everyone's on soft money with the exception of me. So if we take on a student, it's my responsibility to pay their stipend until they're done. And technicians are not state line either. So if we don't get a grant or we lose funding, then we have to send people home. So I think you have to view it as a business. But it's one of the best businesses because you're able to generate, an idea or a thought and then investigate it, right? So it's a privilege. But it's also very difficult. And imagine almost like Shark Tank, right? You imagine you've got this idea and you put it down on paper and you send it out and you go, hey, I've got this idea for a therapeutic for these bacteria and it's going to work. It's going to be fantastic. It's going to save all these lives. Now give me $1 million for it. And maybe it'll work.
I think Shark Tank might be easier than an NIH study, right?
100%.
Absolutely. But I think you have to keep it all into perspective. And it's easy to get, worried because, oh no, what are we going to do about funding? And it's easy to kind of rail against the reviewers, reviewer number 3 on your paper or on your grant. But, it is I do view it as a privileged position because if we can think of it and we can get the funding for it, we can do it.
Absolutely. So we're going to end with this question. We're going to pick your brain or maybe ask you to get out a crystal ball or roll the dice, whichever you choose. Are there pathogens out there that you think we should have on our radar for the quote unquote next big one?
I don't know that we're going to necessarily have the next big pandemic, but I think what we're already seeing is kind of, what's the right word? Reemerging viruses in particular, like dengue virus and triple E. We've seen, big turnaround with West Nile virus. A lot of these vector-borne diseases were wiped out in the southeastern United States with the use of chemicals, right, by spray. Well, we've discontinued that use, and these are making a comeback. And chikungunya virus is another good example in Florida, right, in the Caribbean. So I think those are really, in my view, some of the most concerning because, how do you control that? in a vector. That's very difficult to do.
And we don't have vaccines or treatments for most of those.
Exactly.
Correct.
Exactly. And even, and you know, like dengue virus, for example, we've been working on vaccines for dengue virus for, 60 years, 50, 60 years. And we're still not really that close. And there have been some not so great outcomes with some of the vaccine candidates either. So you have to worry about, you know, things like aberrant immune responses. And this happens with natural infection too. So that's not an easy thing to deal with. So that's what we've kind of turned our attention to, are things like Rift Valley fevers. And Burkholderia pseudomallei is another one that we didn't think that was endemic in the United States. And now we've recently figured out that, actually it is in Texas and Mississippi, and people that have never left the United States are now that we're looking for it, they've been infected with it. So those are two that we've specifically focused on, and we're looking at another round of funding with DARPA for therapeutic and vaccine development for those two pathogens.
Well, we've probably frightened our listeners enough for one
Well, I mean,
episode.
the good news is we're telling people that we're studying these things.
Oh, yeah,
we're not
absolutely.
waiting for it to happen, right? So,
Absolutely.
and I think that's the
And
promise
there's
of
funding.
tomorrow,
Yeah, and there's funding.
right?
So it's the governmental agencies are well aware of this, and I think that's where I'm positioned, myself and others, to do this kind of work, because we have the facilities and we have the trained staff to actually do it.
Well, our guest today, who is standing between us and those dangerous pathogens, has been Dr. Jeff Hogan. He's a professor in the Department of Infectious Diseases and the Department of Biomedical Sciences. Jeff, thanks so much for being with us.
Thanks again for having me.
Links to additional information about Dr. Hogan's research can be found on our Instagram account, which is @PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes launched last season. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Thanks to the New Media Institute at Grady for use of their Studio Not Found podcast facilities, and a special thanks to our production assistant, Sid Wigand, for research, editing, scheduling, and keeping us on track.